Thank you so much, Felix and Liang, for inviting me. I will share my screen. And do you see my screen?
Yes, we can see that. Thank you.
Okay. So I know that on Liang's screen, it says that I'm going to talk about that paper, the most recent paper that we had in Nature Communications. But I decided also to give you a little bit of a brief overview of some of the other works that we do in my group. I'm a computer scientist by training, but we work on genomics in general. And this, for the last few years, we have been doing some cancer genomics. A big focus here is really on single-cell DNA. There's a lot of work on single-cell RNA, but we have been doing a lot of work on single-cell DNA. Why cancer? Not just talking about single-cell in general, it's the issue of intratumor heterogeneity. So this is something that is actually factored into the methods that we develop. So the issue with cancer, as most of you know, is that it is a heterogeneous disease, that when we take a tissue from a patient and we look at different parts of that tissue, we find different mutational signatures in different regions of that. And this is what's known as clonality, heterogeneity, subclones, clones, all sorts of names for the same term. But informally, each clone has certain mutational signatures. And this is, of course, important for the diagnosis and prognosis and treatment of cancer, but from a method development as well, it's important to account for it. I am, as a computer scientist who works on biology, I like to think about biology in the lens of evolution. So I like to look at all data from an evolutionary perspective. Of course, here when we talk about evolution, I put it in double quotes because we are looking now about how the cells divide and differentiate, right? It's not evolution at the species level, but we look at the cells. So looking at it from an evolutionary perspective, if we look at from left to right in this figure, we have these regular tissues, healthy tissues in yellow. Then all of a sudden we have this mutation. The mutation, for example, due to selective sweep or whatever, it takes over the population, this purple mutation. And then, subsequently, we start getting other mutations that start defining their own clones. So if we look at the rightmost side or end of this figure, we have multiple clones in this tissue. If we take a sample from a patient, we have the purple clone, the green one, the red, the orange, and so on. And this is what the clonality is, but if we can look at it from an evolutionary perspective, tracing back the evolution of these mutations to their most recent common ancestor, which is a very, very standard thing we do in phylogenetics as well. Now, there's a lot of interest actually in this community in trying to understand what is the mode of evolution of a cancer, right? When we take data and we'll try to look at how the cells evolved in terms of mutations, which one of these modes here is the mode of evolution? So we have linear, where you basically have a sequence of mutations, each one of them sweeping over the population of cells. So we had in the top left here, we had that brown mutation took over the population, then yellow one came, took over the population, and so on. This gives a linear model, and you can look at from a phylogenetic tree or tree perspective. It gives a certain shape to the tree. That this is actually a tree that we call a caterpillar. It's a very long tree, long and narrow tree. There's the branching mode of evolution. It has truncal mutations at the very, very beginning, followed by multiple driver mutations. And then you have these clones that evolve in parallel. The tree is typically balanced. Neutral mutation is where we have multiple mutations happening with no advantage to them. So the tree is really shallow and very, very wide. We have punctuated equilibrium or punctuated evolution. We have the early stage of tumor defined by burst of genomic aberrations in the beginning. That's why we have that long branch at the tree. So lots of it here. Sometimes you have whole genome duplications. And then you have, after that, a few other mutations that define the clones. Now, understanding this mode of evolution is important for again, diagnosis, prognosis, and the therapeutic treatments. I'm not going to go through this, but here, this is actually a very nice paper that appeared a few years ago that talks about how can you look at these three factors: diagnosis, prognosis, and therapeutic treatment, in light of the mode of evolution of a cancer. So, for example, if you have linear evolution, a single biopsy is sufficient. But if you have branching evolution, where you have a lot of mutations in your cell population, then you need multiple samples so that you have better coverage. Same thing with prognosis and therapeutic treatment as well. So this is an interesting question here. We recently did some work on trying to predict the mode of evolution from single cell data, and we used for this neural networks. They are actually a natural candidate for this one here because it is a clean to a certain extent classification problem. We have these 4 modes of evolution that we are interested in classifying. But in general, the problem that in my group we have been working on is variant calling, tree inference, and all of that from single cell DNA. So here we have a cartoon of single cell sequencing protocol. In part, this is all we do. A lot of this work with the group of Nick Naven here at MD Anderson Cancer Center across the street from Rice University. So the protocol starts with the cell isolation. Then there's a phase of DNA extraction. Because of the low number of cells, there is whole genome amplification, after which the typical sequencing and aligning the short reads to the reference genome happens, from which we do SNV calling, single nucleotide variant calling. And finally, we are interested in a phylogenetic tree or clonal tree or mutation tree, different names in this community. This is important to keep in mind, this protocol, because there are technical artifacts that happen along the way from the cell isolation all the way to the SNV calling that we have to account for. In particular, there are technical errors that happen in single cell sequencing that are ubiquitous. One of them is the allelic dropout. So we have, if the true genotype is heterozygous, for example, we sometimes, due to allelic dropout, we get to see only one of them. So this is called false negative, in the sense that we miss the mutation that is there. We read only one of the haplotypes. There's missing data. It happens as well a lot, where we have a certain mutation is missing completely from the data. And there are false positive, where the true genotype is homozygous, and we end up reading. These are all technical errors in the data. These are not errors of the inference method, but in the data itself, we get reads with the wrong genotype on them. So questions of interest that we have been looking at are, we have this type of data, single cell DNA, what mutations are present, in what order did these mutations arise, and what are the cell populations, what are the clones in this data. And that's where we developed multiple methods. I am not going to go through almost any of them. I'll just say a few words about one of them. So we started with monovar. Monovar is a method for single nucleotide variant calling, and I believe it was the first method developed specifically for single cell data. And then 2 years ago, we did Phylovar, which scales this up to genome, to very, very large data sets with millions of single nucleotide variants. We have SciFit, which we published in Genome Biology. This is a method for building cellular trees from single cell data under what we call a finite sites model. So we allow recurrent mutations and so on. And finally, we have CycloneFit, which is the most recent of these methods. This is a method that works also on single cell data, but accounts for heterogeneity. So CycloneFit is a method that takes as input, single nucleotide variants that have been called by some method. If you look at it, the input here, the matrix has zeros and ones. These are the presence or absence of a mutation. What's colored in red, the zeros and ones colored in red, this is just to indicate that there is error in this data. So CycloneFit doesn't assume the data it takes as input is correct. It assumes that there is error, and it takes care of that in the inference. What the method gives back is the clonal clusters. It tells us, for example, in this cartoon here, if you look at it, you have a tree, and at its leaves, you have 5. It has 5 leaves and 5 clusters. from right to left, again, red, that orange, green, yellow, blue. So it tells us that there are 5 clones in this data. It tells us what are the genotypes of these clones, what are the mutations that define each clone. And then it tells us also the evolutionary tree on which these clones got defined or they evolved. Okay, so it gives all these 3. It employs a very detailed graphical model that takes into account the true genotype that we don't observe. That's a G in this figure, the observed genotype, which is D. But also it makes assumptions about the clonality. It employs something known as the Chinese restaurant process as a prior on the clustering and so on. So it employs this graphical model, and then we use Markov chain Monte Carlo to do inference under this model. I will just give you, and also we account for other artifacts in single cell sequencing, like doublets, where 2 cells can be trapped in the same well, and you end up reading the data from not 1 cell, but 2 cells, for example, at a time. And this was published a couple of years ago in Genome Research. The paper has a lot of results there for evaluating the method, but I want to show you just an example of what you get from this method. So here, this is an example of applying Cyclone Fit to a metastatic colon cancer data set. So we have single-cell DNA data from multiple cells obtained from this colon cancer patient. What you see here in panel A, this is the output of Cyclone Fit. So Cyclone Fit determined that there are 5 clones in this data set. So there's a clone of normal cells, right? A cluster of normal cells. There are the other clusters as well. And what I'm showing you in B, this is taking the output of Cyclone Fit and feeding it to another method called Machina by Mohammed Al-Kabir at Illinois and Ben Raphael at Princeton and others, that tries to predict metastatic pathways for this cancer. But this method requires data or information that comes from a method like Cyclone Fit, where we have to have the clonality that is inferred by this method. We are now working a lot on copy number aberrations. So we have reviews and benchmarking of some of the existing methods for doing copy number aberrations from single cell data, as well as we have a most recent method here that we just published called nested BD for Bayesian inference of these trees from copy number aberrations under a birth-death model. It's not under parsimony. We have a lot of methods under parsimony that try to minimize the number of changes in copy numbers. But this is a Bayesian approach that actually employs birth-death model of gene copies. Okay. So this is a very brief and quick overview of the type of work that we do. And I'll get now to the paper that Liang and Jing invited me to talk about. The method is really very simple. So maybe 15 minutes, I'll be done with my talk, or 15, 20 minutes max. So what is this paper about? It is about integrating single-cell DNA and RNA, and again, in the context of cancer. And the issue here is that there are lots of data sets for which, from the same tissue or the same patient, single-cell DNA and RNA have been obtained, but not from the same cells. So they are taken from the same tissue, but not from the same cells. And it's very important for us to integrate them so that we understand the interplay between DNA and RNA, as well as I will show. Okay. So from this slide, the main point here is just for you to keep in mind that when I talk about DNA and RNA here, I'm talking about every cell is represented by 2 vectors. You know, we have the blue vector, which is the copy numbers. This is at the DNA level. So here, 0, 5, 7, 2, 2. For example, we have partitioned. Imagine we partitioned the genome into 5 bins. On the first bin, we have 0 copies. On the second bin, we have 5 copies of that gene or that genomic locus, 7 copies, and so on. The same thing, we have a gene expression vector that from the RNA that gives us the number of copies of that gene. While the copy number at the DNA level can be applied across the entire genome, So we have copy numbers for coding and non-coding regions. Of course, for gene expression, it has to be for the coding regions. So this is here we are talking about the coding regions. And since these vectors have to be matching, so we need them to be aligned in the sense that really the first entry in each one of them correspond to each other genomically, right? So this is why also the copy number data, when we do this integration, is restricted to regions that have protein coding genes as well. Okay, so this is not the entire genome and non-coding regions. This is restricted to regions with the genes in them. Principle behind this, or an assumption behind this, is something that makes sense to a certain extent, is that potential relationship is that copy number of a gene is higher, the gene is used more by the cell. So in other words, we assume that there is a positive correlation between copy number at the DNA level of a gene, as well and the gene expression at the RNA level. So this is an underlying assumption. It is used it is observed in many studies. It is used in other methods as well. So here again, the issue is that we go back to the intratumor heterogeneity, that we have the way we are thinking about this method, not just for developing it, but for evaluating it, as I will show, that we are assuming that we have healthy cells from which we start having clones of abnormal cells here. Every clone have their own mutational signatures. Okay, so we will assume that there is some sort of correlation between the copy number aberrations and the RNA expression in the orange part, or in the blue part, or in the red part. Okay, so the problem that we are dealing with is really very simple to explain or to define. So we have gene expression vectors and we have copy number vectors. Again, gene expression vectors, imagine we obtain data from 1,000 cells. So it is 1,000 gene expression vectors of integers, okay, positive integers. So if we have read the RNA from 1,000 cells, then we will have 1,000 vectors that have a certain number of entries. And again, each one of them has a non-negative integer. The same thing with copy number. We have a certain number. The number of vectors do not have to match. So you could have 200 cells from which you obtain the RNA, and you could have 800 cells from which you obtain DNA. And our task is really to match them, to match cells from with RNA to cells with DNA. So here, for example, when you look at the right side of the figure, this is, on the left hand, we have the gene expression. So we have, in this cartoon example, we have obtained the RNA or the gene expression data from 3 cells. And we have, on the right-hand side of this graph, we obtained the DNA from 5 cells. So you have 3 on the left, 5 on the right. And we want to match them. We want to match red to red, yellow to yellow, green to green. Now, of course, I'm coloring them here for the sake of illustration. The data we have is this. We don't have colors. We don't know that. If we had the colors and we know which one matches to which one, then we don't have a problem to solve here. The data is given to us. But this is how the data is given. You have the RNA, you have the DNA. We have no idea which cell maps to which cell. Now, please keep in mind that this is a technique we want to apply to data for which we know for a fact that most of the data is coming from, the DNA is coming from a cell, the RNA is coming from a different cell. So the cells from which the DNA and RNA coming are almost disjoint, right? So this matching here, I'm not saying that we are matching the cells as if the DNA and RNA coming from the same cell. No, they are not coming from the same cells, but we want to match them based on some principle that comes really from the heterogeneity in cancer. Okay. So why do we need to do this? I mean, this has to do with the technology that's being used. So there is high throughput sequencing, and there is low throughput sequencing. There are studies where low throughput sequences has been applied, and it does not require integration because they are able to obtain the DNA and RNA from the same cells. So if you are able to obtain DNA and RNA from the same cells, we don't need to run this method. There's nothing to integrate because you know the data, which cells have the DNA and RNA. So the mapping or the matching, in a sense, is given to you. But the issue that this is expensive and all the data sets we are aware of from which DNA and RNA are obtained from the same cell are very, very small data sets. On the other hand, you have the high throughput data or sequencing technology that you have large number of cells from which you have DNA, large number of cells from which you have RNA. They are from the same tissue. But the issue is that we don't know which cell maps to which cell, or what is the correspondence among these cells. And that will require integration that we do here computationally. Okay, so what we really need is an accurate integration method for this high throughput data that enables us to link DNA and RNA from these individual cells so that we understand these correspondences. As I mentioned before, that we assume that the correlation between copy number and gene expression coming from the same clone is higher. So now I'm starting to talk about clone. Again, the important point here to keep in mind is we know that in most data sets this is going to be applied to. These are not coming from the same cell. The DNA and RNA are not coming from the same cell. So we don't want to actually emphasize or stress that condition or put that as a condition in the method, because then it's not applicable to most data sets if we say force them to come from the same cell. And here we use a simple measure of correlation, which is the Pearson correlation coefficient. And this is why I said the method is really simple. What was surprised us is that such a simple method outperformed the more sophisticated methods there. So what we do here is we build what we call a bipartite graph. So bipartite graph, for those of you who are not familiar with the terminology, is a graph where you have nodes on the left side and the right side. So you have some nodes on the left, some nodes on the right. Typically, they come from different types, represent different types. And now you have edges connecting nodes from the left with nodes from the right. So no edges on the left between nodes on the left, no edges between nodes on the right. And here in our case, the gene expression vectors are on the left. So here in this case, we have gene expression data from 2 cells. So we have 2 nodes on the left. We have, if we have the copy number, DNA copy number from 2 cells, that's on the right. Then we match every cell on the left to every cell on the right. So we pair them, that's through the edges. So you see an edge, whether it is in gray or black, between every node on the left and every node on the right. And the final piece of this graph construction is the weights on these edges, and these are the Pearson correlation values. So here when you see W11 is minus 0.1, this means that the correlation between the RNA vector of the first cell on the left and the DNA or copy number vector of the first cell on the right is minus 0.1. Okay, and the same thing for the other. So we have 4 correlations to calculate, and we have 4 edges with 4 values on them. What we are interested in here, so the higher the correlation, the better. So what we said is that we now need to find what we call a matching. So for every node on the left, we want to match it to some nodes on the right, such that the sum of the Pearson correlations on the edges used in this matching is maximized. So if you look at here, for example, if you match the 2 nodes on the left to 2 nodes on the right, there are a few options for that. So if you use the 2 dark edges here, the 2 black edges, you look at the correlations on them, it's 0.3 and 0.9. The sum of these 2 correlations is 1.2. If you take any 2 other edges, any 2 edges from the left to the right, the sum of the correlations on them is going to be less than 1.2. So these 2 edges here, mapping both the RNA vectors on the left to the same DNA vector on the right, this will be the best solution here without putting another constraint that I will mention soon. So this will be the maximum weight edge matching here, or edge cover. But the one other thing we wanted to emphasize or to stress here is that we don't like this. We want to cover all the nodes on the right and all the nodes on the left. So we wanted what we call one-to-one matching. So this one here on the previous slide, we don't like it because the RNA from both cells on the left were mapped to the same DNA on the right. Here, I'm showing you example of the 2 possible matching where we don't allow that. So you map the first one on the left to first one on the right, second to on left to second on the right, or first to second and second to first, as you can see on the top one here. So now what we are interested in is finding such a mapping or matching that has the highest weight. So this is known as the min or max weight edge cover problem here. So this is what we have in this method. So if you give me DNA and RNA, we will build this bipartite graph. It's complete bipartite graph. So we will link every DNA vector to every RNA vector. We will compute Pearson correlation coefficient on them. And then we will find this matching that matches every RNA vector to a DNA vector. Okay. Now, this is a better illustration of the method because there are some other details in the method that I am just gleaning over here. So if you start from the left to the right, again, the data is the gene expression matrix and the copy number matrix. As you can see on the columns, they both have the same number of columns. So you have 6 genes and in both of them. Okay. So we have that. And then on the number of rows, they don't have to have the same number of rows, because the number, again, as I mentioned, the RNA could be obtained from more cells than from DNA or vice versa. From that, we build this bipartite graph here. So we have the RNA cells on the left. I refer to them RNA cells as in the cells from which we have RNA. Okay, and when we have the DNA cells on the right, we calculate the matching. So we have already built the edges with correlation coefficients. Now from this, we will do another thing. So if you look here in this bipartite graph, you'll see that there's more RNA than DNA. So one detail that's in the paper that I didn't show in the previous slide, what happens when you have more RNA to DNA? You have no choice but to connect or to match more than one RNA cell to a DNA, right? So the way we do it in a heuristic manner. So first we do a phase where if we have 6 cells from which we have DNA, so we do matching of 6 cells from which we have RNA to them. Then we remove these 6 cells from RNA and we repeat. We keep repeating until all cells from which we have RNA are mapped to the cells for which we have DNA. Now, there is the next step that we do here. So if you look at the bottom here, we do clustering or clonality. So imagine that we run cyclone fit on the copy number data, or you run hierarchical clustering or whatever clustering method. So here what we get, for example, in this cartoon, we get 3 clones or 3 clusters. This is done based on the DNA data. Okay, so we do the clustering or the clonality detection based on the DNA data. Once we do this, now, because we know the mapping of the RNA to DNA from MaCroDNA, we map them also to the clones. So now we start looking at it at the clone level. Did we map the RNA to the right clone, not to the right cell? Again, I want to go back to that point. Cell-to-cell mapping doesn't make much sense for us to say this is the right cell because they are not the same cell. We know that for a fact. But we want to map the cells of the RNA to the right clone, right? So we are assuming if there is clonality at the DNA and RNA level, the clonality should be similar. And what we are saying here is that let's evaluate or let's look at these methods at the level of clonality. Did we map the RNA to the right clones? And this is what you see here. So we have 3 clones on the right, and then we have these orange circles of the RNA. So we have 9 cells from which we have RNA. 3 of them are mapped to the blue clone. 5 of them are mapped to the orange clone. 1 of them is mapped to the green clone. So this is really the overall of the method and how we are going to evaluate it as well. So we're going to say, did we map them to the right clone or not? In terms of existing methods, there's Clone Align, which was developed specifically for integration of DNA and RNA. And there is Surat, which was not developed specifically for DNA and RNA, but it is really a manifold method for integrating of data from different modalities. Okay, so you could apply it to DNA and RNA, but it was not specifically designed for DNA and RNA. Okay, so when we evaluated the method, we compared it to these 2. There's a third method. It is mentioned in the paper. The results are in the supplementary material. we didn't get any good results from the method. We did not include it in the main text, but it is in the supplementary material for those of you who are interested. So one interesting thing in this study is that we evaluated the methods on data for which we know the ground truth. So if you look at this data, so from this paper here, it was published in Science a few years ago, they had data on multiple patients from which they obtained single-cell RNA, DNA, but also for many of the patients, they had DNA and RNA from the same cells as well. So we focused on only 3 patients here, CRCs 4, 10, and 11. If you look at every row, we are showing you the number of cells, individual cells from which they got DNA or RNA or both. So for example, in patient 4, they had RNA from 93 individual cells. They had DNA from 93 individual cells, but they had 57 of these 93, 57 cells, they had both DNA and RNA. So this is now a very interesting data because we can run these methods on these data and say we know the ground truth because the data was obtained from the same cells. So we know the ground truth here. The same thing with patient 10, there were 69 cells from which DNA and RNA were available, and 174 cells from which DNA and RNA were available. Now, this is important to stress here because when we submitted the paper for review, the reviewers were insisting on simulations. And in my own work, I mean, we do simulations in lots of papers, but I just didn't know how to do simulations in this context here. How do we simulate DNA and RNA from the same cells? It was a challenging thing. So this is why I was very happy about finding this data because this is data, biological data. It's not simulated for which we know the ground truth. But for simulations, if anyone here can help or knows of some tool that realistically models or simulates DNA and RNA from the same cell and at the individual cell level, we would love to look into that. So here are some figures of the evaluation. Here I'm showing you the cell to clone accuracy. What does this mean? Here we mean is that we know the ground truth. So we know which cells, which RNA should map to which clone because we know the ground truth in this data. And now we are looking at each one of the methods and saying, what was the accuracy of that method? How often did they map things correctly? The measure is between 0 and 1. So 0 means nothing was mapped correctly. 1 means 100%. All the RNA was mapped to the correct clone. Okay. Now, in addition to clone-align and Surat, we also ran baseline method. What is the baseline method? We took an RNA for every cell for which we have RNA. We mapped it to a random cell for which we have DNA. So it is a random method, just randomly assign things and see how that performs. So if you look at the left plot here, you see that MaCroDNA. So we have three shapes there, right? The star, the X, and the plus. They correspond to different patients. Each one of them is summing over multiple cells there. And you can see, if you look at the star, MaCroDNA is the best. If you look at the circle, and if it's the plus, and you look at the X, MaCroDNA is also the best. We also log transform the data because it is right skewed, and it is very important to look at log transformation of that. You see that MaCroDNA is still the best performing. The others' performance actually drops down significantly. We were surprised by clone-align. It didn't do well, in particular for log transform data. It basically did not map any cell correctly. For the untransformed data, for one of the patients did very poorly. The others one was okay, one was very, very poor as well. One interesting thing here is to look at the baseline. So this is just a random assignment. If you look at it, in fact, on the untransformed, you see it's not far from Surat and from clone-align. In fact, for some of the patients, it's better than clone-align. One of the issues there is that the clones themselves were unbalanced. Okay, so there was a very large clone that a large number of the cells map to, and this just impacts this when you start randomizing things. If you have 99% of your data belong to one large cluster, then even random assignment is going to work just by chance. We experimented with rebalancing the clusters by sampling from them, and the results are shown in the supplementary material of the paper. But again, if you look at the right panel here, Also under the log transform data, still MaCroDNA is the best. This is here about correctness of the clone and the assignment. Did we assign the right cell to the right clone in terms of RNA? This is a different measure here. We looked at the clones and we look at the prevalence of these clones. So for example, one clone, we know in ground truth for one of the clones, 50% of the cells there have RNA that should map there because we know the ground truth. So that will be the prevalence on the x-axis here. So when you see 0.4 on the x-axis means the truth tells us that there is a cluster where 40% of the cells have RNA mapped to them, okay? On the y-axis here is that the prevalence based on the prediction. So the diagonal line is perfect performance, that cluster that had 40% of its cells having RNA. Also the prediction mapped, or the methods mapped RNA to 40% of the cells there. As you can see here, again, based on this figure, clone align and the Surratt are the farthest from the diagonal. MaCroDNA, which is in purple, is the closest to the diagonal. So if you look at these measures, in terms of the prevalence or the balance or the number of points in a cluster, or if you look at the identity, which cells were mapped to which cluster, in both cases, log transformed and untransformed MaCroDNA performs best. In addition to this, which I'm not showing on the slides, but it's in the paper, there's a lot of pre-processing that had to happen in the data, and there could be multiple ways of pre-processing the data. We played with that as well, and there's lots of extensive results in the supplementary material where we have similar figures, but for different transformation, different selection of genes, different pre-processing of the data as well. As I mentioned, the method is very simple. We were surprised by how well it performed, and it is also very efficient computationally. So if you look here, this is on the top row is the time consumption, the amount of time in seconds. The bottom row is the amount of memory in gigabytes. The left column is untransformed, and the right column log transformed. If you look at it, the top curve is clone aligned. That's the slowest of them, followed by MaCroDNA all in terms of the time. When we restrict the the data or the processing of data to 2,000 genes, we get the bottom as well. Okay, so if you look at the memory usage, Surratt and clone align consume the most amount of memory. MaCroDNA is the best. When you look at running time, all of them are better than clone align. But also, if you restrict the data set to about 2,000 genes, so do all that analysis based on 2,000 genes, not on all genes in the data, also the amount of time for MaCroDNA and Surratt becomes comparable as well. One other thing we did here is that we said, okay, now this was validation, looking at the data with ground truth. Could we do something interesting with this method? And the fact that the method allows us to get to map RNA to DNA, we wanted to look at, can we use this method now, or the mapping obtained by MaCroDNA, to understand this correlation between the mutations at the DNA and RNA level. So then we looked at this data set of Barrett's esophagus, where data were obtained from different patients, multiple patients, at different stages, healthy or non-dysplastic or low-grade or high-grade, and then cancerous. Okay, so there were 4 stages in that NDBE, the non-dysplastic Barrett's esophagus, the low-grade LGD, and the high-grade HGD. And then there was the esophageal adenocarcinoma, the AAC. And we wanted to see, because the data from different patients, each with a different grade here, we wanted to see, can we see something, glean something from the data that tells us, is there any indication in the data, DNA or RNA, that can distinguish these? Because in the original study from which the data was obtained, they couldn't, from the data they obtained, DNA or RNA, they couldn't tell anything that was distinguishing among these, especially for the high-grade and cancerous ones. They couldn't distinguish them from the others. So there were no signatures in the data that allowed them to say, well, here are some indications about why this patient is at a certain grade. So we took the data from this and we applied the MaCroDNA. Again, this is data here from every patient. They obtained DNA from some cells, RNA from other cells. In the original paper, they analyzed them independently. Now that we have MaCroDNA, we said, let's integrate them and do some analysis on them. So we took every patient, we took the data, the single cell data, RNA and DNA. We ran MaCroDNA on that. It gave us matching, and the same thing for all patients. Then what we did is what we call phylogenetic signal analysis. So what is phylogenetic signal analysis? Phylogenetic signal analysis, we use the DNA data to build a phylogenetic tree, an evolutionary tree of the cells. So imagine here you have 5 cells. You build an evolutionary tree for that. Here in our case, we used a simple method called UPGMA. We built a tree. Once you build the tree, at the leaves of the tree, you have the DNA vectors or the genotype vectors from which we build this tree. Now, MaCroDNA allows us now to map the RNA to the leaves of this tree, because MaCroDNA tells us which RNA vector maps to which DNA vector. The DNA vectors are already at the leaves of the tree. We used MaCroDNA to map them to the leaves of the tree. Now you have the leaves of the tree have the RNA on them. We did not want to build a tree from RNA because I don't know how to do it. I don't know what is a model of evolution for RNA. For DNA, we have models of evolution from which we can build the trees. So MaCroDNA, using the DNA, we build the tree. Using MaCroDNA, we map the RNA to the leaves of the tree. Then once you have the RNA at the leaves of the tree, you can do analysis. Actually, we used a package called PhyloSignal. It's not ours. It's actually a method that is published in the data that measures the correlation between the RNA and the tree shape. So if you look at these two hypotheses here on the left and right, and we are putting some extreme examples here. So the values you are seeing here are the gene expression levels. So 6, 3, 2, 102, 221. If you look at it here, what happens in the left, the cells in which you have a much higher RNA level, they are grouped together on the tree. Whereas on the right, the 102 and 221 are in different branches of the tree. So in a sense, it's very hard to say that there is a correlation between DNA and RNA if what you see is the figure on the right, because the mutation at the DNA level are the signal for building the tree. Okay? The mutations at the DNA level are the signal for building the tree. Well, if there are mutations at the RNA level, they are not following the tree shape because 102 and 221 are very far from each other. Whereas on the left, they are closer together. So if you have these two hypotheses, using this PhyloSignal, there is a measure called the K or K star, it will tell you if the data resembles the one on the left, so there is high correlation between that. If it's like the one on the right, it will say there's very low correlation between the signal in the DNA and the signal in RNA. So we use this kind of technique and we applied it to the data here. Again, when you look at every panel corresponds to one patient, and what's above the panel is the stage. Again, NDBE is the non-dysplastic Barrett's esophagus, but on the rightmost column is the high grade and the esophageal adenocarcinoma. Okay? What we found here is we use this technique. What we found is that for the high grade and the esophageal adenocarcinoma, the correlation between the DNA signal in the tree and the RNA is much higher than the other grades. Okay, so if you look at these figures here, and in each one of them, the inset there is we are zooming on the tail after K star of 1. And you see here that there are larger numbers of points of higher than 1. So I don't want to jump to biological conclusion here. The data could have issues in it, but this is just an illustration, one illustration of what you could do with this integration. You could use the DNA to build evolutionary trees or cellular trees, or do intratumor heterogeneity analysis to get the clones or whatever. You can then use MaCroDNA to map the RNA to the DNA. Once you are done with that, now you can start looking at, is there any signal between the heterogeneity or the tree structure and the RNA? Now, any correlation you find has to do with the DNA because the tree structure is inferred from the DNA itself. And the same thing with the heterogeneity. So this was just an illustration of a biological data analysis where we're not looking at the accuracy of the method here, but we were looking at, here is some nice example of what you can do once you integrate this data. Okay. So to summarize here, for MaCroDNA, it integrates single-cell gene expression and DNA, and it outperforms existing methods in terms of accuracy. Again, if you look at the paper and supplementary material, we looked at the methods in many ways. We also, for clone-align in particular, because we were surprised by its performance, we did very thorough analysis to make sure we are fair to the method and we were not just, reporting bad results. But you see all these results in the supplementary material where we tried to play with all its parameters and to see if we could improve it. One limitation at this point of the method is that it is a combinatorial method. It does not provide confidence intervals or uncertainty. It says, here is the matching. It does not say, here's the matching and we have 90% certainty about it. And that's something we are working on to develop, to allow us to also quantify certainty in the results we obtain. All the methods I described, including MaCroDNA, are implemented, publicly available for the community to use, to run, to modify, as well as to give us feedback as well. Okay. And with that, I want to acknowledge all the students and the collaborators who have been working on this. This has been done a lot with Ken Chen and Nick Niven, both faculty members at MD Anderson Cancer Center. And all the RICE people are my former and current students as well. And on one of the projects, we corresponded with colleagues from Spain as well. And with that, I thank you for your attention, and I'm happy to take any questions.
Well, thank you. That was so inspiring, and thanks for taking the time to present two independent stories. So for everyone, feel free to put the questions into the chat for the Q&A. We have about 10 minutes for the speaker. Let me maybe just start with one question, and that might just be like a more big picture question at the intersection of computational biology in the clinics. So moving forward in this field, when you think about it, like where we might stand in the next 5 years, is there anything that we can learn here that might be of therapeutic or diagnostic value? I know a lot of the techniques obviously cannot be implemented at a clinical point of care, but would there be anything that we could extract that could potentially be implemented into either how we care about patients or even how we might change our therapy in the future?
Yeah, thank you very much, Felix. I mean, this is a very good question. It is a very broad question. As a computer scientist, I've been in this field for about 20 years, 25 years. I always tell my students that they should never make a claim that they are curing cancer. We are computational people. We will never cure cancer. The way I view computational work is really the importance of the work is to narrow down the hypothesis for the experimentalist, for the biologist to work on. So, for example, rather than saying, the genome has 30,000 genes, go to experiment with each one of them. If we can come up with a computational method that will narrow that down to 500, 500 is better than 30,000. This is how I view it. Now, one thing that I am intrigued by with this kind of work, when I talk to colleagues at MD Anderson, for example, so I was speaking to a colleague who works on pancreatic cancer, such a devastating disease. There are some who believe, maybe this is actually has been established in the literature, I'm not sure, but believe that, even pancreatic cancer, maybe the mutations that lead to it started 20 years before the onset of the pancreatic cancer. One thing that of interest to me is that can we using these methods, zoom in on biomarkers, on certain mutations, on certain diagnostics that we could do at certain point, through blood tests or whatever, that allow us to tell 20 years before that, there is a propensity here for pancreatic cancer. Because these methods, what we do with them, when you look at them, these are evolutionary methods. They are reconstructing the past, right? So they are reconstructing the past. Yes, we are looking at data from today. The cancer patient showed up today. They took the tissue, they sequenced it. These methods now are going to give us the story going back to the most recent common ancestor of this. I would love to get to a point, can we time them Can we time them to see when did this start? At what point? I am not a cancer biologist or cancer genomicist. I'm actually a phylogeneticist. I have been working on phylogenetics for a long time. We do that type of work in phylogenetics. In phylogenetics, when we build methods, not only do we build the past, but also sometimes when you have fossil data, stuff that helps us calibrate these trees and get better estimates of time. Now there are more and more patients where you have data from the same patient over multiple time points, sometimes before, after treatment, sometimes, people are showing to the clinics and getting blood work, throughout their life. Maybe we can zoom in on some of these to try to time these mutations and hopefully get to some tests that allow us to do something. And there was a slide that I showed, something about diagnosis, prognosis, and stuff, and the mode of evolution. It's still, I would say it is still academic there. But if you think about it, if you have clonality there, you might want to apply the treatment in such a way that you shrink certain clones in this data if they have bad mutations, especially bad mutations, and so on.
That is a very detailed and visionary answer. Thanks so much for that. And I appreciate it was also a very broad question. So great. Maybe like a more computational question. So you mentioned a lot of your models are inspired by graph theory. And I think one of the advantages could be that they are very explicit and actually interpretable. So I was curious whether that might be an additional advantage that you could maybe elaborate on, because I know interpretability, the more we move toward machine learning models, is often an issue. They might perform very well, but we don't even understand why they are performing that well. And I think, like, for instance, the bipartite graph models that you showed could be actually something that might perform well, but also be interpreted why they have this accurate performance.
Absolutely. I mean, Felix, you are hitting on very important points. Now everyone, the buzzword is deep learning and large language models, and everyone wants to throw these things at every problem. I don't apply them unless I am convinced that they make sense. I'll give you an example where they make sense to me. So we do a lot of inferences in phylogenetics where the likelihood function is a nightmare. We cannot even write it mathematically, let alone compute it. Now, if I could use, deep learning to solve the problem for me in a likelihood-free manner, that's not bad, because I don't know how to do it in a likelihood-aware manner, right? But when it comes to something like this, I completely agree with you. This bipartite graph, when you get the matching, it's not you're getting not just a number. You are going to actually tell you which cell maps to which cell. Then you can zoom in on that and say, where did we get that value for the Pearson correlation coefficient? Which are the top genes that are driving this value as well? I think in biology, in general, we need interpretability. There are certain areas that sometimes also interpretability gets thrown left and right unnecessarily, right? If you are running a method that's doing hill climbing search in a large space, and I could replace that by deep learning somehow, that is not about interpretability, because how do we interpret even, hill climbing, right? I mean, it is just a heuristic working in the space. So if I replace hill climbing by Markov chain Monte Carlo, or Markov chain Monte Carlo by a black box that we call a neural network, I don't think that's an issue of interpretability. It's completely fine to run it there. But if we are trying to see, for example, in the case of Barrett's esophagus, if we are trying to find which are the mutations at the DNA or at the RNA level that will allow us to tell the difference between the different stages, I don't know then what it means for us to use, a deep learning or neural network to say, you will be in this stage or you will be, but what is it that we should be doing, right? Going back to my example, it's not sufficient to have a black box that tells me in 20 years I will have, again, I'm using hypotheticals here. It's not sufficient to have a black box that will tell me in 20 years I will have pancreatic cancer. It's not helpful. What do I do now for 20 years, right? So I'm a big fan of interpretability. I think I have one paper that has used deep learning. Again, there was a reason for using it there. It was really to replace some search. It's not about the biology, it's about the computation.
That makes a lot of sense. That brings me to one kind of related question that we have. And essentially, like, comparing like different methods in different types of scenarios. So you mentioned a lot of your work has been related to colon cancer. So I was wondering, what would applications look like where the ground truth might not be that well known, like either rare cancer types or cancer types where we might not even have the correct annotations, like what type of cancer it was, or where the diagnosis would be uncertain?
Yeah, so we're developing the method for these scenarios specifically, Felix. We did not develop it. I mean, when we applied it to data where the ground truth is known, that was just for validation. How are the methods working? That's how we were able to use the word accuracy, right? Because we know the ground truth. But we are developing the method for biologists or clinicians or who want to apply it on data there's no ground truth. In fact, to data where they know for a fact the DNA and RNA coming from different cells. That was the very last bullet point in my slide where I said, our method now does not allow us to assess accuracy or certainty in this. So that's something we want to work on to develop, is that in addition to saying, here is the matching, but also it will attach to it some sort of uncertainty value there, right? So, imagine you could do bootstrapping, you could do in a Bayesian setting, you could get the posterior distribution of solutions rather than here is one solution for that. So that's something we are looking into now.
That makes a lot of sense. Thank you so much. And I think we're almost exactly at the hour. Liang, do we have any last minute urgent questions?
Yeah, I'm not sure whether we have time. We have actually one question in the Q&A.
Yeah, do you want to quickly read?
Yeah, so there's one question. Well, it's very great talk. Thanks. And so the question is, when pairing RNA and DNA, do you see a big influence in the sequence depths on the DNA side? Have you checked it?
Yeah, that's a good question. This is why I mentioned in my talk at one point that we did a lot of pre-processing on the data. These are all biological data. So if you look at the paper, especially in the supplementary material, actually in both the main text and supplementary material, we talk in detail about how we pre-process the data. So yes, you have to pre-process the data. For example, clonal line, if I remember correctly, like it doesn't accept, you have to remove every gene that even one cell has copy number 0 in it. So if you have one gene and there's a copy number 0, you have to throw that gene, or you throw out that cell, right? One of the two. So yes, I mean, at the end of the day, I mean, here we are taking the method takes as input copy number profiles. They have been calculated. So back to the question is that if the data that is poor, the sequencing data is poor, and the inferences are going to be maybe problematic. So now the input to MaCroDNA is problematic. Currently, we do not account for that. We don't, like for every node for which we have DNA, we are not also putting another measure of how accurate this copy number profile. This has been estimated by a method, okay? But that's a great question. And on the pre-processing, we did it. Did we evaluate the performance of the methods, under different reads and stuff like that? No, we didn't. But another issue to account for is, how do we run this method if we put also some measure of certainty around the copy number profile or the RNA profiles, because these have been estimated computationally as well, right? So the data itself already has error in it on the left and right of the graph. Yeah.
Well, it's a perfect answer, and I think a great way to wrap up today's session. So it was wonderful meeting you virtually. Hope we'll have the chance to catch up in person at some point when you're coming to Boston later this year. Thanks again for letting me co-host the seminar, Liang. And yeah, looking very much forward to our upcoming speakers then.
Thank you very much. I'm honored for being invited to give this talk. Thank you so much.
Thank you so much. Thank you both.
Thank you. Thank you so much again.